So let's hope this is as interesting for you as it is for me. Okay, so I'm not one of the authors of this work. It's from my research group. Since none of the authors could be here, I've been asked to present on their behalf. So to paraphrase Ben from a few days earlier, please don't ask any hard questions. But that said, I think this is really interesting work, and I'm excited to talk about it. So it's about how to uncover new drivers of cancer by more accurately mapping somatic mutation rates. And what are somatic mutations? Those are the kind of mutations you acquire during your lifetime. So despite the immense progress made in understanding these mutations during and how they contribute to cancer, a sizable portion of tumors still don't have a known driver mutation. So here, for example, I'm showing you non-small cell lung cancer, and about a third of the tumors don't have a known driver mutation, which leads to the question, where are all those missing drivers? And one hypothesis, a big part of this particular talk, is that they are hidden in the non-coding genome. And for the longest time, we only used to do whole exome sequencing. Now we are starting to do whole genome sequencing for tumors. So we're starting to get a better sense of how this comes about. The second hypothesis is that there are driver mutations of the kind that we know and see in some cancers, but they are rare in other cancers. So there is this long tail of driver genes, which are commonly mutated, but we don't see them. So essentially what happens is if you look at standard or like the common mutations, they occur frequently enough that you can measure them and you can see them. But then in other cancers, some of those mutations might be so rare that we don't have the statistical power right now to be able to tell them. So this work is in that direction. And to understand how to identify these drivers of mutation, I think it's useful to think about how somatic mutations come about and how they are sort of selected for. And throughout our life, we acquire these somatic mutations. Occasionally, one of them gives a proliferative advantage to the cell. It grows, it has clonal expansion, and then once in a while, that leads to cancer. Now, the key point is that this mutation is giving a benefit to the cell. So this is under positive selection pressure. And so then this kind of a mutation shows up over and over again in cancer as you sequence a bunch of different tumors. And so you end up sort of seeing that as a driver mutation. And so this, for example, is the story you would see with something like the gene TP53. You see it recurrently mutated across samples. The problem is a high mutation rate by itself is not sufficient. So for example, a gene like Titin shows up with a fairly high mutation rate all the time. But it is not a driver mutation, and its mutations do not provide a fitness advantage to the cell. So the resolution to this mystery, and a bunch of people have noted this, for example, Lawrence et al. In 2012, is by looking at the number of mutations you should be expecting at a neutral mutation rate. So the point is that Titin just seems to be liable to have a lot of mutations. And that's why you see that. And those are not necessarily driver mutations. And so one useful way of sort of trying to look for driver mutations is to look for cases where in cancer you see the mutation at a much higher rate than you would expect from the neutral mutation rate. And while this particular example is about genes, the same intuition applies to any part of the genome, to the non-coding region as well. So that is the key idea of this work. They present a highly accurate genome-wide model of somatic mutation rates. You can use this model to then do a test for signals of positive selection in cancer and see, okay, what are the rare driver mutations that might have been missed. Now, the idea of modeling variations in mutation rate is not new. That has existed for a while. The contribution of this work specifically is that rather than focusing on a specific, say, genomic feature like a gene, they are trying to do this at scale across the entire genome simultaneously. And so how do you do that, right? In a coding sequence, you can look for synonymous mutations and use that as a proxy for neutral mutations. But in the non-coding genome, the 98%, you can't really do that. You have to do something else. So one clever observation that Max and Adam had was essentially that it has been noted that epigenetic features in healthy tissues are highly predictive of somatic mutation rates in cancer. And there are some reasons for this that people understand. So for example, if it is compacted chromatin, you end up seeing higher mutation rates because the repair machinery has relatively little access to the DNA. The thing is, we don't understand all the mechanisms of how this happens, but what this study did was sort of step aside from that, like why it's happening, and just say, can I use this? And so what they did is they designed a deep learning method that leverages epigenetic data from roadmap epigenomics to predict somatic mutation rates at kilobase-scale resolution. And now this epigenetic data contains a lot of different things. It contains histone modification patterns, DNA accessibility, methylation, and so on. And I'm not going to go into a ton of the detail. For those who are interested, they had a paper in ICLEAR 2021, and then there was a follow-up in Nature Biotech. Those have all the details. But I will give some broad intuition. So broadly, what they do is they assume that if you have some region of the genome, the mutation count is coming from some negative binomial distribution. And their goal is to model its mu and sigma, the mean and the variance. And so they get to that by essentially by essentially using a Gaussian process to predict that. And the epigenetic features form the input to that Gaussian process. So the way it starts, you start with some resolution parameter, and you can run this model at different resolution parameters, the minimum being, I think, 100 base pairs, but this particular example here has, I think, 10 kilobases. So for a particular region of the genome, you can think of it as a sequence, and they have access to 735 epigenetic features. So you can think of it as a sequence with 735 channels. That is put into this convolutional neural network, and the resolution parameter controls sort of the kernel size of the convolution. And the end result of this convolutional neural network is a small fixed length embedding. And the idea is that you take all of those epigenetic features and you collapse that into a small latent space. And that latent space essentially ends up being the input to your GP, which then predicts what the mu and the sigma of the negative binomial will be. And so in sort of the way I have understood this is basically the idea is you use the epigenetics to kind of characterize the region of the genome, whether it's an enhancer, a TSS, or whatever else, and then you use the GP to kind of come up with the parameters of neutral mutation rate. And the final thing is that you have a prediction for mutation rate everywhere along the genome. And so I'm showing you this here at a 1 megabase resolution. So in the blue is what this method predicts. They call it DIG. In black is what is the actual mutation rate. So at a 1 megabase resolution, their method is explaining about 98% of the variance, which is pretty cool. And the thing is that because they have this flexibility of zooming in and out onto any resolution, they can almost make it look like a genome browser. And so what they ended up doing is they made browsable maps for 37 cancer types from the PCOG dataset, and you can explore that at that URL. So now that you have this background somatic mutation model, you can use that to test cancer-specific mutational spectra and see if any of those mutations are showing evidence of positive selection. So basically, the test is very simple. You essentially look for excess observed mutations against this expected neutral background rate. And you can do this for all kinds of things. You can look at coding mutations. So for example, in NRAS, you can look at mutations in the promoter or the non-coding regions. So here I'm showing you TERT. And interestingly, you can also look at mutations in introns, and so look for splice variants that end up happening. And this is VHL. So I'll give you a few vignettes of their work. So the first vignette is around cryptic splicing. So the idea is that there is a mutation that happens in an intron that leads to a splice change or a deleterious impact on the protein function. And so what they did is they essentially applied their method to all the introns, did like a splicing test, and they looked for individual genes in specific types of cancer with an excess of intronic cryptic splice single nucleotide variants. And what they found seems to be fairly consistent with what is known. So for example, for SMAD4 and TP53, which are drivers across numerous types of cancers, they found cryptic splice SNVs across a pan-cancer cohort. So that is sort of in the bottom right. But then for something like PBRM1, which is a kidney-specific tumor suppressor gene, they observed the cryptic splice SNVs only in kidney cancers, so which kind of makes sense. And so before this work happened, there had been some efforts to sort of try and identify non-coding region mutations, and people have had difficulty doing that. But Reinway et al. In 2020, they found one, for example, in the 5' UTR of TP53. So in this work, Max and Adam found more support for that finding, and then they went and basically looked for mutations in the 5' UTRs of other genes. And they found a few. One of the ones here is the 5' UTR of ELF3. And there's some good evidence here. So for example, in this, let's see if I can use this. So this here, the point is that there are two different data sets, PCOG and the Hartwig. And they were able to find evidence for positive selection pressure in both data sets for this 5' UTR SNVs compared to other places. So that makes them somewhat confident that this is a reasonable thing. Of course, I mean, there could still be some neutral mutation explanation for this, but this seems plausible. Then this last one, I think, is fairly broad analysis and really showcases what the power of this kind of an approach would be. So what they did was they identified individual genes with an excess of activating or protein truncating mutations in specific cancer types. And here in gray, they are showing the specific gene-tumor pair association. So this is a fairly busy figure, but the top block is looking at oncogene and tumor pairs. The bottom block is looking at tumor-suppressing gene and tumor pairs. So along the x-axis, you have genes. Along the y-axis, you have tumors. So essentially, the gray is indicating that that particular gene is being associated with that tumor. And the size of the gray indicates the size of the association. And kind of the point being made here is that for something like an oncogene, you are surprised to see that this box is fairly sparse. You're not finding associations for oncogenes with certain kinds of tumors, but only with other kinds of tumors. And why is that? And that, in some sense, was the motivation for this work. And so they did a broad analysis, and they said, essentially what they found was that there are associations for these oncogenes and also tumor-suppressing genes across a variety of cancers. So for example, they were able to associate AKT1 with essentially all the 10 types of cancers here, or ERBB3 with 7 types of cancer. And the point is that the landscape of common and rare driver genes likely overlaps considerably. So essentially, an oncogene in a particular tumor might be common versus rare, but it might still be a driver in both cases. And it's just that we haven't had the statistical power to find the rare ones yet. But this method starts to get to that in a bit. And as we get more data, hopefully these matrices will continue to grow and be filled in. So with that, I'll summarize. I think the key takeaways here are, one, this work builds an accurate genome-wide model of somatic mutation rates. It enables efficient search for mutations under positive selection. And a couple of findings. One is that cryptic splicing might be a bigger deal than is being appreciated. So it might be a significant driver of mutations, especially in tumor suppressor genes. About 5% of candidate driver SNVs might be from cryptic splicing. And the other part is that somewhere between 1 to 10% of tumors, there probably are rare driver mutations coming from well-known oncogenes or tumor suppressor genes. And this, for example, might have therapeutic implications because you might be able to use medications designed for that particular oncogene in a cancer where you typically don't think it to be active. So with that, this is work by Adam and Max primarily. They both have graduated and are doing a startup. And then Puru and Bonnie. So yeah, with that, I'd be happy to answer any questions I can, but don't keep your hopes up.